Hi guys, so today we will be studying the concept of supplementary genes, right? So like in complementary genes, you needed two dominant copies to show you a phenotype. In this case, you have one gene which will originally give you a particular phenotype, right? So for example, if I take color in any, let's say maize, or even a very good example would be rat coloring as well. So for example, for a rat to be brown in color, it needs the pigment melanin. Similarly, for a maize grain to be red in color, it needs a pigment which will give it a red coloring, correct? If that particular gene which gives you that red color is present, then obviously you would get the red color in maize. Or the gene responsible for melanin production is present, then you would get a brown colored rat. So that's plain and simple. If it is not there, then it's a white colored maize grain or it's an albino rat. But in this case, because again we are studying non-Mendalian inheritance, you have a second gene which when present will modify the expression of your original present first gene. So here one of the best examples that we can take is maize. So we will do the cross with maize obviously. So in maize you have a white grain which is the double recessive type. So the genes R and P, the R giving the red coloration is in recessive state. So you get a white grain, plain and simple. If it is a red grain, then obviously the R has to have one present dominant state allele. So you have one capital R and the second gene is recessive. So no effects whatsoever. In the third case, however, when the second P gene is present even in an heterozygous state, you get what? You get a purple grain. Similarly, if we want to think about it in a simple term, then rat, let's say gene A is responsible for the production of melanin and gene B is the supplementary gene. So if gene A is present and gene B is not present, then you have a brown rat. But if gene B is present in a heterozygous condition, then it promotes or accelerates the production of melanin and you end up getting a black rat. Right? So when we look into the biochemistry of it, the idea is that there is initially a white or a colorless pigment which is giving you the recessive genotype or the white grain, correct? If there is the R gene present in even one dominant copy, then obviously the enzyme R will get produced. The white pigment will get converted into the red pigment and you will get the red grain, correct? If now, however, the supplementary gene, the P gene is also present and is expressive, then it will convert the red pigment into a purple pigment and then you will get the purple product, which is the purple grain. That is the concept of supplementary gene then, when one particular gene at a particular locus will supplement the effect of the primary gene at a different locus, correct? I hope this was clear to you. Thank you.